Hello and welcome everyone. We're gonna just get us started as everyone is still joining. Thank you for joining our webinar titled Next Generation Bioprinting Platform, a multimodal approach for bioprinting of cartilage, multicellular spheroids and skin. This is Nilufar Khosravi from Syntica, and I'm pleased to be your host today. At Syntica, we provide enabling technologies to advance your research. Our portfolio includes a physiology, imaging, and biofabrication line of products. That includes Poetis and NGB-R that we are going to talk about it today. Our session's speaker is Dr. Fabien Guimont, the co-founder, CSO, and president of Poetis, a biotechnology company um, based in France, specialized in development and manufacturing of living human tissues by 4D bioprinting. Fabien is an entrepreneur with over 2 decades of experience in regenerative medicine, holding a PhD in material sciences, specializing in health and life sciences. In 2005, he was appointed as a researcher at INSERM in France and became an invited speaker at Harvard University in 2010. He's pioneering in bioprinting and he's an inventor with 100 plus publications and patents. Fabien has enhanced his scientific expertise with entrepreneurial training from HEC Paris and founded Poetis in 2014. In this webinar, you will discover the next generation bioprinting platforms that we call them NGB platforms, which uniquely combines the benefits of laser-assisted bioprinting lab technologies, such as high resolution and improved cell viability with bioprinting methods like bioextrusion, and this integration facilitates the creation of intricate multicellular 3D constructs. You will learn about also the features such as robotic automation and the fluidic devices within the platform that effectively bridges the gap between laboratory bioprinting and clinical application. We will showcase this translation capacity through bioprinting of skin and cartilage spheroids for building larger tissue structures. We anticipate that today's presentation by Fabien will take about 25 minutes, so we will have time to answer your questions live. Please feel free to submit your question on the Q&A dialog box all throughout the presentation. And please note that we will be recording today's session. You will have access to the video file to review at own time after the webinar. So before we start the presentation by Fabien, we have 5 quick poll questions to better understand the interest of the audience. And if you could please take a moment to answer them, that would be appreciated. I'm going to read through the questions. So question number 1, are you currently using any 3D bioprinting technology? The answers would be yes or no. I start to see answers. A few more seconds. Question number 2 is what organs or tissues are your primary target for bioprinting? Options are skin, vital organs such as liver, kidney, heart, lung, pancreas, or vascular tissues, bone, tumor, other, or none. Question number 3 is what applications are you exploring right now with the bioprinting technology? Select all that applies to you. You can put multiple choices. The options are 3D cell culture models, lab-on-a-chip devices, organoid/spheroid bioprinting, disease modeling and therapeutic testing, or you use it for basic research/education. 4th question, what is the main challenge you're facing right now in adopting bioprinting technologies? the cost of it, lack of expertise or training at your facility, limited available material, or other. The last question would be, how soon does your organization plan to acquire or upgrade to bioprinting technology? So within 6 months, within 12 months, within 2 years, or no plans for now? We're gonna give a few more seconds for everyone to put their answers there. Okay, awesome. Thank you so much, everyone, for your responses and your feedback. With that, I'll hand it over to Fabienne. Fabienne, thank you so much
Yes.
for joining us today. Please feel free to start your presentation when you're ready.
Yeah, thanks, Nelufa, for the introduction. So that's my pleasure today to present you what we are doing on what the technologies that we have developed on its first application in skin and cartilage. So just briefly, so Poietis is located in France, in southwest of France. So we have been established in 2014. Our mission is to provide solutions to both research centers and regenerative medicine centers, and we commercialize namely 2 types of bioprinter. The one for research is called NGB-R, and the one for therapeutics fabrication is called NGB-C. And we in parallel to this commercialization, we developed some tissue therapeutics with a first focus on skin, namely on Poie skin, which is a an autologous dermoepidermis substitute. So we have been developing for the last years what we call the next-generation bioprinting platform. I guess that you are all familiar with extrusion, which is the conventional technique that is used in the field of bioprinting, and that is pretty useful to print 3D shapes containing cells on biomaterials. The matter with these technologies is that it's very difficult to control the internal architecture of the tissue, since the distribution of cells within the tissue is basically derived from the local cell concentration within the syringe. Also, the resolution of this technology is quite coarse, and is basically defined from the needle diameter. If you reduce the needle diameter, you increase shear stress and unfortunately increase cell death. That's the reason why we have been developing the NGB platforms that enables keeps the capacity to print 3D tissue shapes, but unable to control the internal architecture of the tissue and to control the microenvironment of cells by providing, thanks to the capacity to deposit patterns of different cell types of ECM with a very high resolution, and patterns can be anisotropic, non-geometric as in native tissues. Basically, thanks to this NGB platform, we can deal with tissue complexity, and we consider that bioprinting becomes now driven by the biology principle. So the need to control the internal tissue architecture to promote cell-cell interaction and interaction of cell with extracellular matrix component at the cell level. So this NGB platform is based on a unique technologies that we have developed at first at INSERM, which is the National Institute for Health and Medical Research in France. So this technology is called laser-assisted bioprinting. Its principle is pretty simple. So we use laser pulse to focus these pulse. These laser pulse are focused on a cartridge, a glass cartridge that is transparent to the laser light. And by focusing this laser pulse at the interface between the glass slide and the liquid film that contains cells, that has been spread onto the glass slide, we can generate some very small jet of liquid and thus created very small droplets that contain a given number of cells. And interestingly, one pulse generates one droplet, and we can fire with the laser at more than 1,000 pulses per second, meaning as many as a droplet on the substrate per second. The main advantages of this technology is its high resolution, around few tenths of micron, and since it's a nozzle-free technology as compared to extrusion or inkjet technique, for instance, it provides a very high cell viability, even for quite fragile cells. And we have shown that it is compatible with a large amount of tissue components. It could be hydrogels, living cells, but also spheroids. So that can be used also as building block, as you will see later in my presentation. So the technology is very versatile in the sense that we can print solutions that has a quite high range of viscosity from 1 to 1,000 centipoise, so meaning very liquid to quite highly viscous solution. Also, just by tuning the main parameters that are namely the laser pulse energy, we can print either cells in suspension, cell aggregate, or at the opposite, exosomes that are used now in many regenerative medicine applications. And we can print also different kind of a range of concentration of cells from less than 1 million cells per ml to even very concentrated suspension of cells reaching 100 million cells per ml, for instance. So we have worked a lot over the last years to automatize laser-assisted bioprinting. So now our system are now equipped with some microfluidic cartridges that are used to, for instance, print multiple cell types that are been first positioned in different reservoirs. So we combine, for instance, this microfluidic cartridge with this an automated pipette that is positioned in a 6-axis robotic arm to print different cell types and create complex multicellular constructs. So as an overview, so the NGB printers are basically a combination, a multimodal system on combined laser-assisted bioprinting head with the microfluidic cartridge in combination with what you could find in other bioprinters, namely bioextrusion on UV print heads. And we combine these two printing technology with other technology, namely high resolution inline microscope. So that has a resolution of 2.2 microns. And interestingly, this microscope can be used to, for instance, monitor what you have printed, but also thanks to the view print function, enable to print precisely onto a specific location of your substrates. For instance, if you use a lab on a chip or organ on a chip with specific channels, for instance, you can go to the microscope and select a specific area of these chips to print then specifically the pattern of cells or extracellular matrix components that you want. So it's pretty useful and open new avenues in terms of application. So all these technologies have been integrated in a biosafety cabinet that also enable let some free space for instance, preparing your bioink or do the cell culture and so on before or after the bioprinting. So on this system is coupled with different software. At first, Zebra 4D which is our CAD software for tissue design, on a NGB interface that is used to edit your protocol on execute the printing process. So Zebra very quickly has this structure. So it's basically divided into 3 panels. So at first, it is used to define the 3D shapes. For instance, you can import a STL file, for instance, and then the 3D shape is filled with different layers of bioinks that can be printed either with laser bioprinting or extrusion. So basically, when you design your tissue, you fill from the bottom all the layer defining the parameters on the pattern of cells, for instance, for each layer. So it's pretty intuitive and very powerful and very really dedicated to printing complex tissues. So the machine is also coupled with this NGB software where we have 2 menus basically. One is for editing. On there, you define the printing parameters. You define, for instance, the type of labware that you want to use and so on. And when you have defined and set your protocol, you go to the printer section to execute the bioprinting. So as an example, we have also another software that is used with the NGB-C bioprinter, the clinical grade bioprinter. For it, it's a bit the logic it's a bit different, since it's the goal of this software is to execute SOPs, standard operating procedure, and it is used namely to execute the process. The operators have to check different boxes so that the protocol is performed according to what has been developed at the preclinical stage. So as a commercial point of view, we have started the deployment of the printers last year. At first, we installed the first bioprinter in the first half of 2023, and installed the first bioprinters in the US early this year. And we have also started the commercialization of the NGB-C for clinical application in different cell therapy centers in Europe. So in the US, as you know, Cintiqa is our distributor. You can contact them if you are interested in having a NGB bioprinter. So in terms of application, as said before, we have developed our focus on the skin substitute market, which is a growing market with some very large medical needs due to the aging population, the incidence of several comorbidities like diabetes, obesity, cardiovascular disease. What we also observe as well is a growth of the advanced wound care market that use more and more advanced skin substitute like skin epidermis and so on. So on the goal from many interviews performed with clinicians and plastic surgeons, we defined that the goal was to find or to develop an alternative to the standard of care, which is today the split thickness skin graft or autograft that has strong limitations in terms of availability at first, but also the final aesthetic outcome are not satisfying. There is a lot of wound contraction and thus some pain for the patient. So in that perspective, we have been developing Poieskin, which is our first therapeutic product. So Poieskin is a 3D bioprinting autologous dermoepidermal skin substitute. On its indications are the treatment of burns, acute and chronic wounds, but also reconstructive surgery. So from the interviews we made with clinicians, we defined the target product profile. On thus, Poieskin is a 40 square centimeter skin substitute, 8 by 5 centimeter with a stratified epidermis already competent for grafting at the day of grafting. On the expectation from the clinician is that the grafting can occur about 3 weeks after the biopsy. So, and we have developed a process that is compatible with this time frame. And the expected benefits, as I mentioned, is to overcome the limitation of photographed, and also hopefully to reduce the wound care expenses that are pretty high. So we developed a specific process based on leveraging the multimodal capabilities of the NGB platform. So at first, after extracting the cells that are human dermal fibroblasts and keratinocytes, after the expansions, we go to the printer. And at first, we print the dermis by printing collagen, bovine collagen, on fibroblasts to make the dermis compartment. And after a few days, we go back to the printer to print keratinocyte to make the epidermis on the top. Afterwards, the construct is positioned in airlift condition to promote differentiation of the epidermis. And interestingly, as we have been able to automatize a lot the manufacturing process, reducing the fabrication, the bioprinting duration, at least for the dermis, to less than 4 hours now to make this 40 square centimeter. What is really important for the cell therapy facilities that we produce Poieskin for patients in the future, because you have to envisage that the manufacturing process is not too long for operators and for these facilities. So we obtained some nice dermoepidermal skin substitute with, as you can see there, well stratified epidermis with all the different layers of the epidermis, from the basal layer to stratum corneum on the top, namely with anatomopathologist from Marseille Hospital. We've developed a scoring method to evaluate the quality of our tissues. And basically that will be in the future some that will provide some release criteria to make that this tissue can be implanted into patients. So we also evaluate the efficacy of Poieskin in vivo using nude mice. So, and we perform that in comparison with human tissues. And interestingly, at first, that was also a criteria given by the surgeon. We have demonstrated the suturability of Poieskin, which is important for clinical practice. On in vivo efficacy results, we observe a very similar graft take of Poieskin as compared to human skin, on similar contraction profile and so on, which is really promiscuous for the future, since our goal is now to move to a clinical trial that will be probably one of the first of its kind in the field. And for that, we have performed tech transfer to Marseille Hospital, installing a bioprinter there, and we have trained the operators so that they can make and fabricate Poieskin. So we are now on the production of the batch in the GMP condition with the hope to start a clinical trial in the close future, and that depends on funds, as you may understand. So in parallel to this skin application, we are also expanding the application of a platform namely on cartilage. And this is projects that we perform with in the context of a consortium funded by the European Commission. So this project is headed by the Catholic University of Leuven in Belgium, that has a very old experience and expertise in the field of cartilage. And the goal of this project is both to develop some osteochondral implants fabricated via the bottom-up assembly of skeletal stem cell aggregates that are previously differentiated into functional tissues. And the second objective is to design and develop robotic platforms that combine bioreactors, in-process control on bioprinting, on specific bioprinting technologies. So in the context of this project, we have namely developed what we call the PIC cell module that is used to print spheroids. So basically, for instance, here is a schematic, for instance, of a cartridge where some spheroids are positioned. So we use a specific system to image the cartridge, and thanks to a specific algorithm, segmentation tool, and so on, we can select, for instance, by the size, a specific aggregate that will be transferred afterwards onto the substrate. So, for instance, here is some first results that have been published quite recently in Biofabrication Journal. So you see there some aggregate onto the printer. So we select them using by image processing, basically, and afterwards we can arrange these aggregates according to very specific patterns. So we have shown that this module is biocompatible and can be used to print, for instance, larger construct made of aggregates our goal is to increase now the size of this implant to be able to implant them into animals. So the conclusion, so the bioprinting platform is the NGB bioprinting platform. It's one of the most advanced bioprinting system in the field. So it's a robotized system with different technology, laser-assisted bioprinting, bioextrusion. Interestingly, it meets unmet needs of tissue engineering, ensuring the standardized fabrication on the capacity of printing multiple cell type makes that we can print 3D complex and functional tissues. And hopefully in the future, our goal is to be able to install this printer as well at the point of care directly in hospital to make that hospital cell therapy facilities can print this tissue and provide more affordable treatment for patients. So I would like to thank you for your attention, and we'll be happy to answer questions.
Thank you so much, Fabian, for the interesting and insightful presentation. So we have a few questions on the dialog box and also in the private chat, so I'm going to go through them. Could you please explain more in detail the process, mostly in the part with air liquid interface? I assume it's referring to the skin biofabrication process.
Yeah, so as I said, when we first print the dermis and then print the epidermis, and then the construct is at first culture in immersed condition to ensure the proliferation of organization of keratinocyte, the first layer. And then we put the construct in air liquid condition, so meaning that the dermis is positioned in immersed condition into culture media, while the epidermis is positioned in air. And this promotes the differentiation of the epidermis progressively. And that is how we obtain a stratified and functional epidermis at the end. So this culture in air liquid condition is quite common, I would say, for epithelial tissues on epidermis, namely.
Okay, thank you so much. So the next question, again, related to the skin biofabrication process that you probably already answered, why you wait a few days before between printing human dermal fibroblasts, the dermis, and then the keratinocytes on the epidermis?
So we have tried different conditions, for instance, printing the same day, the fibroblast on the keratinocyte. What we find is that waiting a couple of days for maturing the dermis is provide a better anchoring of the epidermis. While if you as compared to a construct where you have not wait the maturation of the dermis. This is probably due to the remodeling of the dermis that occur at first without the keratinocyte. So we also during this time there is also some proliferation of fibroblasts. It's remodeling plus proliferation of fibroblasts. So at least from an experimental point of view, we observe better results so far with this few days of maturation of the dermis before printing the keratinocytes.
Well, thank you so much. The next skin-related question, do you ever have issues with basal keratinocytes not having proper polarity? Have you tried integrating a basement membrane to help mitigate this issue?
Yes, it's a key question for everyone who wants to create some skin tissues. Indeed, we have we had issues for that. The maturation of the dermis is one of the answer. Also, there are some specific culture media that also promote the maturation of the dermis that at the end ensure a good polarity at the dermoepidermal junction of the good polarity of the keratinocytes. So we have not tried to integrate another component by, for instance, we know that some proteins are present at the dermoepidermal junction. Also, because we are in a perspective of performing a clinical trial on when you add a lot or when you add component, it makes the tissue on the process more complex. And for clinical applications, usually the simple is the best, I would say.
Thank you so much. Next question is regarding the aggregates bioprinting. Have you considered or tried using organoids rather than spheroids?
Yes, indeed, is what we are starting to do in another project funded by the European Commission that deal with brain. Our goal is to print mini brains based on organoids. So it's something that we are just starting now. I would say that in principle, the principle is the same, is based on this pixel module.
Okay, great. Are you considering using machine learning / AI for improving the printing process? You already mentioned you are.
Yeah, indeed. We already do that with the pixel module, but our goal is also to leverage the AI capabilities for other purpose. And interestingly, our platform is a robotic platform that already integrated a number of sensors with a microscope and so on. And our goal is to use all these sensors to generate data to be able to use or to leverage AI capabilities afterwards to improve the resolution or to make the result more predictable, I would say.
Okay, great. So another question posed one of the audience. What approaches would you take to print vascularized tissues?
So it's something that we are, for instance, we are also dealing with in the context of the projects for printing mini brains. So we currently print some endothelial cells in combination with mesenchymal stem cells or pericytes by doing co-patterning or co-printing of both. We try to create some vasculature of capillaries is the main approach that we have been developing so far in the context of vascular bioprinting.
Okay, great. Next question. What are the commonly used biomaterials with the lab technique, laser?
For laser-assisted bioprinting, as I said, the technology is pretty flexible in terms of viscosity. And usually cells are basically only suspended into their own culture media. So you don't need to mix them with specific hydrogels or biomaterials. But for certain application, we integrate some biomaterials in combination with cells. Typically, we use either collagen or alginate. It's probably the two main materials that we use internally, at least.
Okay, thank you so much. And the last comment from one of the attendees: as an emerging company, are you looking forward to expanding your team? I guess yes, always. Please feel
Yes,
free to
always.
yeah,
I would say.
feel free to reach out to any of us. We would be happy to have a conversation on the chat. Oh, just another question came in. Any interest in the cardiac pathology field, like cardiac patches?
Yes, indeed, as well. We namely partner with an hospital in Paris for that. And in that context, this project is not based on printing aggregates. It's basically the opposite. We are much more interested in printing exosomes. So that makes that all the projects give us new capabilities, printing cell suspension, biomaterials, aggregate, and now exosomes. So it's a quite new project for us, but it will present some very promising applications as well.
Okay, great. Seems like we have answered all the questions. And thank you so much, Fabienne, and everyone who attended our session. So we will share the recording of the webinar with you. And for any questions or inquiry, please feel free to reach out to myself or anyone from our team. My email information is on the slide, or just simply send an email to info@syntica.com. And thank you so much. Have a great rest of the day.
Bye-bye. Thank you.